Good morning, everyone. This is Dr. Veena K. from Maharani Science College for Women. This is course BSc for 6th semester, subject Chemistry, Paper 8. In this session, I'm going to discuss about Introduction to Metabolism, that is what is catabolism, anabolism, and in detail about carbohydrate metabolism. So, whatever we eat, whatever we start chewing, putting into the mouth, the process of digestion starts. So what happens in this process of digestion? Which are the steps involved? How the food what we eat, whatever may be the form, is converted into energy is known as metabolism. That is what we are going to study in metabolism. So metabolism is nothing but converting food into energy. So metabolism is a process which converts whatever the food we take in into energy. Whatever may be the food we take in, that is converted into energy in the process called as metabolism. Now, the sum total of all biochemical changes or the biochemical reactions that take place in the living organism is termed as metabolism. And in this, there are two types. One is anabolic reactions and then catabolic reactions. Remember, anabolic reactions requires energy, which is also known as the synthetic pathway, because the smaller biomolecules are combined together to give a big biomolecules in anabolic reactions. In catabolic pathway, the bigger biomolecules are broken down into smaller ones with the release of energy. So, in catabolic process, the bigger biomolecules are broken down into more simpler compounds. Therefore, delta G is always negative, and here delta G is positive, and here the more complex compounds are produced from simpler molecules. So, look here. Catabolism is the set of metabolic pathways that breaks down the bigger biomolecule into smaller ones with the release of energy. Okay, and anabolic processes, look here, the smaller units are combined together to form a bigger molecule with the help of energy. So, anabolic reactions requires energy and also known as endergonic process. Now, see, there are different types of biomolecules we come across. They may be carbohydrates, lipids, proteins, vitamins, minerals, chlorophage, water, etc. So, before I come to carbohydrate metabolism, whatever the food we take in different forms, it may be a carbohydrate, it may be a protein, or it may be a fatty acid. In the first stage of catabolism, all of these are undergo conversion into the simple units. If it is a carbohydrate, it breaks down into a monosaccharide. If it is a fatty acid, it breaks down into smaller units of fatty acid. Okay, that is simple units. And then if it is a protein, it is broken down into amino acids. Okay, you know very well, large number of amino acids combined together to give proteins. Similarly, if it is any fatty acid, they are broken down into glycerol and then fatty acids, smaller molecules. Now, in the second stage of catabolism, all this, the simpler units of carbohydrates, proteins, or if it is an oil or a fat, they are broken down into fatty acid and then glycerol. And in the second stage of catabolism, all of these are converted into acetyl coenzyme A. So the beauty of this metabolism is whether we take a carbohydrate, a protein, or a oil or a fat or lipids, they are broken down into simpler units. And in the second step, they are converted into a common intermediate acetyl-CoA. And this acetyl-CoA enters into Krebs cycle and then TCA cycle follows. So these are the three stages of catabolism, right? Now, coming to carbohydrate metabolism. In this session, I am going to mainly deal with the carbohydrate metabolism. You know, carbohydrates are the major source of energy from our diet. They are composed of carbon, hydrogen, and oxygen, and also called as saccharides, which means sugars. Carbohydrates are polyhydroxy derivatives of aldehydes or ketones, and they are commonly known as saccharides. Now, see, these are a simple unit of sugar. We call them as monosaccharide. When the two monosaccharides combine together, look, we get disaccharides. When large number of monosaccharides combine together, what we get is a polysaccharides. Now, coming to carbohydrate metabolism, if I use the word carbohydrate metabolism, what you are going to study under this is glycolysis. Glycose means sugar, lysis means splitting. So, splitting up of a sugar is known as glycolysis. It is also known as EMP pathway, named after the three scientists, Embedon, Merov and Paranas, and this is also known as EMP pathway. So what happens in glycolysis is sugar splits, right? How it splits? Into how many steps? So carbohydrate, in carbohydrate metabolism, what we talk is entirely the splitting up of a sugar. Now, glucose is a simple carbohydrate which contains 6 carbon atoms. So in glycolysis, a 6 carbon compound glucose is broken down into a pyruvate. Pyruvate is a 3 carbon compound. So glycolysis ends up with breaking up of 6 carbon compound glucose into 2 molecules of pyruvate. Okay. Now, in this glycolysis, there are 10 steps which are enzyme catalyzed, which converts glucose into pyruvate with a simultaneous production of ATP. So we'll take one by one what are these steps now. Okay. So in the later stages, there is also a formation of lactate from pyruvate here, and the glycolytic sequence of reactions differ from species to species only in the mechanism of its regulation. Okay. So in aerobic organisms, glycolysis is the prelude to citric acid cycle and etc. So it is a central pathway for glucose metabolism. Come on, we'll study glycolysis now. See, if you understand this slide, then the entire glycolysis is understood. So, as I told in the earlier slides, glycolysis is the splitting up of a sugar which converts glucose into a pyruvate 2 molecules, which involves 2 phases. Which are those 2 phases? One is the preparatory phase, number 2 is the payoff phase. So, glycolysis involves 2 phases, preparatory phase and payoff phase. Each phase involves 5 steps or 5 reactions. So, glycolysis comprises of 10 reactions with 2 phases, preparatory phase and the payoff phase. Now, as a student of chemistry, you should know how to write the reactions of glycolysis. So, I'll show in the next slide. It is better you have to understand writing the conversion of each from step 1 to step 10. What is the reactant and what is a product? See, this is a Harvard structure of glucose. Numbering starts from here 1, 2, 3, 4, 5, 6. Numbering is important because every time when the conversion is taken place, you should know on which carbon atom what functional group has disappeared and which functional group has appeared. Okay. So let us begin the reactions of glycolysis in the preparatory phase. See, the preparatory phase, as I told, there are 5 steps. All this is 1 to 10 number are the names of enzymes here. So 10 steps, 10 enzymes, and these are the products or the conversion taking place in each step. So now coming to step 1 of glycolysis, in step 1 of glycolysis, glucose is converted into glucose 6-phosphate. So I'll teach you how to read this flow chart. See, in step 1, glucose is converted into glucose 6-phosphate. I told you this is an enzyme. So which is an enzyme that you are seeing here? Hexokinase. So you have to understand like this. Glucose is converted into glucose 6-phosphate with the help of an enzyme hexokinase. With the help of an enzyme hexokinase. So now As I told, this is the structure of glucose. See, this is the sixth carbon atom. You don't have any phosphate group here. Glucose is getting converted into what? Glucose 6-phosphate with the help of an enzyme hexokinase. And to introduce this phosphate group, we require ATP. That is, this is the ATP requiring step where ATP, one molecule of phosphate group is transferred from ATP to glucose. So further in turn, ATP is converting into ADP. Okay. So in the first step of glycolysis, glucose is converted into glucose 6-phosphate. Now, coming to the second step, how do you read again here? Glucose 6-phosphate is isomerized or is converted into fructose 6-phosphate with the help of an enzyme phosphoglucoisomerase. So, isomerase is an enzyme which converts glucose 6-phosphate into fructose 6-phosphate. You come here. So, this is your glucose 6-phosphate. Is converted into what? Fructose 6-phosphate. So coming to the numbering of carbon atoms in fructose, start from here. 1, 2, 3, 4, 5 and 6. So remember, in the second step of glycolysis, glucose 6-phosphate is isomerized into fructose 6-phosphate. Come to the say there is no ATP or ADP involved here. Okay, this is simply an isomerization reaction. Come to next step. Fructose 6-phosphate is converted into fructose 1,6 bisphosphate or biphosphate with the help of an enzyme phosphofructokinase. I repeat, fructose 6-phosphate is converted into fructose 1,6 biphosphate or bisphosphate with the help of an enzyme phosphofructokinase. See, this is fructose 6-phosphate. I told you this is the first carbon atom. This ATP is utilized for a transfer of a phosphate group. ATP gives one phosphate group, further converts itself into ADP. See, where does this phosphate group comes? On the first carbon atom. So you can see here on the first carbon atom and on the sixth carbon atom you have phosphate group. Therefore, the name of this compound is fructose 1,6 bisphosphate. Right? So in the up to step 3 now, there are 2 ATP molecules are utilized in glycolysis. In the next step of glycolysis, this fructose 1,6 biphosphate or fructose 1,6 bisphosphate is split up into 2 compounds. See, this is a 6 carbon compound. Is split up into 2 3 carbon compounds. Which are those 2 carbon compounds? One is dihydroxyacetone phosphate, abbreviated as DHAP, and the second one is glyceraldehyde 3-phosphate with the help of an enzyme aldolase. So aldolase is the enzyme which converts fructose 1,6 biphosphate into 2 3 carbon compounds. They are DHAP, dihydroxyacetone phosphate, and G3P, that is glyceraldehyde 3-phosphate. Now, whatever the reactions that take place down the line of G3P is true for down the line of DHAP. Okay, so the last step of glycolysis is this dihydroxyacetone phosphate and G3P can be interconvertible with the help of an enzyme triose phosphate isomerase. Okay, so these are the 5 steps of preparatory phase of glycolysis. So up to here, up to the preparatory phase of glycolysis, there is no synthesis of ATP. Remember, there is only utilization of 2 ATP molecules. So if I want to calculate the energetics up to the preparatory phase of glycolysis, I'll take it as minus 2 ATP. Okay, now we'll come to the payoff phase of glycolysis. See, since these two are interconvertible, we are going to study the reactions down the line of G3P. Remember, that is true for down the line of DHAP also. That's why I have written 2 molecules, 2 molecules, 2 molecules products down the line of G3P. So the step 6 of glycolysis is this glyceraldehyde 3-phosphate is converted into 1,3 bis phosphoglycerate. See, this is a glyceraldehyde 3-phosphate, a 3-carbon compound. So CHO group, it's an aldehyde group is there. So this is glyceraldehyde 3-phosphate is converted into 1,3. See, start numbering from this is glyceraldehyde 3-phosphate. This is first carbon atom according to IUPAC name, the functional group 1, 2, 3. 1, 2, 3. So this is glyceraldehyde 3-phosphate. This is converted into 1,3 bis phosphoglycerate. See, at the first carbon atom there is a phosphate group here. First carbon atom main bantu, you have got a phosphate group, and here 2 NAD plus is required with the which undergoes reduction to NADH and then H plus. And see, the enzyme is glyceraldehyde 3-phosphate dehydrogenate, which is a very complex enzyme. This is a very complex enzyme which converts G3P into 1,3 bis phosphoglycerate. Now, step 7 of glycolysis. This glyceraldehyde 1,3 bis phosphoglycerate is converted into 3-phosphoglycerate. You look here. This is, see, there are 2 phosphate groups here. Okay. One phosphate group is given to ADP. One phosphate group is given to ADP. So ATP synthesized. So this is a step where a molecule of ATP is synthesized. So totally there is a generation of 2 molecules of ATP when 1,3 bis phosphoglycerate is converted into 3-phosphoglycerate with the help of an enzyme phosphoglycerate kinase. So please remember, phosphoglycerate kinase converts 1,3 bis phosphoglycerate into 3-phosphoglycerate. See, there is no phosphate group here at the first position. It is only at the third position. So this is 3-phosphoglycerate. Coming to step 8 of glycolysis. See, this 3-phosphoglycerate is converted into 2-phosphoglycerate. Shift of this phosphate group to this position 2. So 3-PG is converted into 2-PG. 3-phosphoglycerate is converted into 2-phosphoglycerate with the help of an enzyme phosphoglyceromutase. Okay. So in the next step, this 2-phosphoglycerate is converted into phosphoenol pyruvate. Phosphoenol pyruvate by the help of an enzyme enolase, where the dehydration takes place. See, this H and this OH. This OH gets eliminated by the help of an enzyme enolase. So when you remove this H and then OH, there comes a double bond. There comes a double bond. Okay. So what you have is phosphoenol pyruvate, PEP, phosphoenol pyruvate. See, in the next step, this phosphoenol pyruvate with the help of an enzyme pyruvate kinase is converted into pyruvate. See, this phosphate group is transferred to ADP and in turn 2 molecules of ATP are synthesized. So this is also an ATP generation step. So in this step, phosphoenol pyruvate is converted into pyruvate. So we started with glucose. We came across the preparatory phase, splitting into 2 compounds, DHAP and then G3P, and this G3P is finally converted into pyruvate, right? 2 molecules. So a 6-carbon compound glucose is converted into 2 molecules of pyruvate. Pyruvate contains 3 carbon atoms, 1, 2, 3. So these are the reactions of glycolysis, which takes place through 2 phases, preparatory phase and phase, via 10 reactions, and these are the 10 enzymes that are involved in glycolysis. Now you have understood sugar has split, or a glucose molecule, a 6-carbon compound is split into 2 molecules of pyruvate. Now we'll see what are the energetics of this reaction. Okay. Now, so as I already mentioned, there are 2 phases. One is the preparatory phase and the second one is the pre-op phase, which glycolysis leads to the breakdown of a 6 glucose 6 carbon compound glucose into 2 molecules of 3 carbon compounds pyruvate, which is bifurcated into two phases. Phase 1 preparatory phase, phase 2 is the payoff phase. Now, so preparatory phase consists of 1 to 5 steps and yield fructose 1,6 biphosphate. This fructose 1,6 biphosphate is then split into two molecules of 3 carbon compound glycerol 3 phosphate and dihydroxyacetone phosphate. Now, first step results in the cleavage of hexose chain. Okay, so remember, preparatory phase requires an investment of two ATP molecules to activate glucose and cleavage of glucose into 3 carbon compounds. Okay, see, these are energy required reaction. Glucose 6 carbon utilizing how many ATPs? 2 ATPs converted into G3P, which contains 3 carbon atoms. How many moles? 2 moles. 1 glucose split into 2 molecules of G3P by utilizing 2 ATPs is the energy requiring reactions which are taking place in the preparatory phase of glycolysis. The same thing. Whatever I explained now is again put in this slide. Okay. Now, I have already explained payoff phase. This is what happens in the payoff phase. So, your glyceraldehyde 3-phosphate is ultimately converted into 2 molecules of pyruvate. Okay. So, the same reactions which I have explained. Now, coming to the energetics of this glycolysis. See, from 1 molecule of glucose, okay, when it's converted into 2 molecules of pyruvate, so ultimately you get 2 pyruvate, 2 ATP and then 2 NADH. Okay. Now, 2 molecules of ATP are generated can directly be used for doing work or the synthesis. See, in the reactions we saw there, 2 NADH are liberated. See, this NADH which has come again enters into ETC, okay, electron transport chain. For 1 NADH, 3 ATPs are synthesized. So, from 2 NADH, we are going to get 6 ATP molecules. 6 ATP molecules. So, remember, under the aerobic condition, pyruvate is catabolized further in mitochondria through pyruvate dehydrogenase and citric acid cycle, where all carbon atoms are oxidized to what? Carbon dioxide, and the free energy released is used in the synthesis of ATP, NADH and then FADH2. Okay. See, under anaerobic condition, what happens? This pyruvate is converted into lactate. Okay. So, this under anaerobic condition, this pyruvate converts into lactate. Okay. Now, coming to energetics of this glycolysis pathway. See, ATP is formed in which steps? Glyceraldehyde 3-phosphate converting into 1,3 bis phosphate, bis biphosphoglycerate. There are 6 ATPs are released. And 1,3 biphosphoglycerate converted into 3-phosphoglycerate, 2 ATPs are released. And then phosphoenol pyruvate converted into enol pyruvate, there are 2 ATPs are released. Remember, these are the ATPs that are obtained from NADH. Okay. So, totally 6 plus 2 plus 2, there are 10 ATPs are generated from the payoff phase of glycolysis. But remember, in preparatory phase, 2 ATPs are consumed, as I have already told. So, glucose, when converted into glucose 6-phosphate, 1 ATP is utilized. Fructose 6-phosphate converted into fructose 1,6-phosphate, 1 ATP is utilized. So, I am going to minus these 2 ATPs from this 10. So, 10 minus 2, 8 ATPs are released. So, what you have to remember from this discussion is, carbohydrate metabolism is nothing but glucose metabolism. It's a splitting up of sugar. So, when 6 carbon compound glucose is split into 2 carbon compounds pyruvate, which contains 2 molecules of pyruvate containing 3 carbon compounds. Okay. Now, it undergoes through 2 phases, preparatory phase and then payoff phase, and the total net ATP released per molecule of glucose when undergoes this conversion of glucose to pyruvate is 8 ATP. Total number of ATP is 8 ATPs are generated. So, remaining, now you have come at the stage where glucose is converted into pyruvate. In the next session, we will see what happens to this pyruvate. So, remember now, Glucose is converted into pyruvate with a net release of 8 ATP per molecule of glucose. Thank you.